So far, we have gone through the appraisal of the common types of articles you will encounter in medical literature, namely therapy, diagnostics, prognosis, harm, and clinical practice guidelines. The last type of article we will discuss in this EBM course is a systematic review and meta-analysis. You already know this, but let's do a quick review. Systematic reviews are distinguished from traditional reviews by the use of systematic methods to identify, select, critically appraise relevant articles, and collect and analyze data from included studies in order to minimize bias. A systematic review may also use statistical methods to summarize the results of included studies. If so, it is referred to as a meta-analysis. Researchers conduct systematic reviews and meta-analyses for the following reasons, which I will not go through anymore, as most are addressed in a supplementary video I provided. You can pause this video to go through these reasons, which may perhaps be of particular importance to you if your MedRes 3 research project involves SRMAs. As with other appraisals, we make use of the PCOM or PECOM models when appraising SRMAs. Basically, a systematic review summarizes the results of several studies that seem to address the same clinical question, with the same components, namely, the patient population with a certain disease or condition, the intervention or exposure, that is, the treatments, tests, or causative or prognostic factors being evaluated, the corresponding comparisons, which are controls, gold standards, or the absence of causative or prognostic factors respectively, the outcomes of interest, be it occurrence of a disease, of complications, or of some other measure of health, and lastly, methodology. When meta-analyses started taking shape in the late 1970s and early 1980s, statistical analysis were limited to measures used in randomized controlled trials. Over time, however, improvements in analyses have been made to accommodate observational studies. Now, as with any other type of appraisal, we follow the same process where we start with appraising directness and end with individualizing results of the study to our patient. Again, when appraising directness, we assess how well the PCOM model in the study corresponds to the PCOM of our clinical scenario. Basically, if you see that the article can provide a reasonably direct answer to your clinical question, then proceed to read it in more detail. Otherwise, search again for a better one. Up until now, you notice that the criteria for inclusion or exclusion have been focused on individual patients. With systematic reviews and meta-analyses, though, we describe criteria for inclusion or exclusion of individual studies. These criteria usually include the methodological quality of the studies to be included, as well as the populations, exposures, and outcomes of interest. Now, as I mentioned earlier, SRMAs have classically been associated with pooling of results from RCTs, but over the years, it has adapted to include other study designs such as cohort, case control, diagnostic, and cross-sectional studies. Now, the type of study design included in the SRMA depends on the type of clinical question being addressed. You see this in the methodology section. It is also here where you can examine the exclusion and exclusion criteria that were used, and should specify the components of the PCOM or PECOM model. Now, keep in mind that the best study design may not always be available for a certain question. In such cases, reviewers may decide to include study designs of lower validity. Now, the next question to be answered under validity appraisal refers to whether the search for eligible studies was thorough or not. A thorough search for published literature should include use of electronic medical databases such as Medline, Embase, and the Cochrane Library, to name a few examples. Check as well if cross-references in original publications were also looked into when gathering published articles. Of course, it is always good if a comprehensive search was made and that unpublished articles, including preprints, have been retrieved and reviewed as well. Unpublished trials can be found here. Unpublished material can also be retrieved by writing to experts, going through pharmaceutical industry files, or surveying conference proceedings and books of abstracts. Now, a very helpful tip to help you answer this question is by going to the methodology and looking for a description of a computerized search, as well as a hand search of relevant journals, personal communication with known researchers, including drug companies, and other methods of searching for articles both published and unpublished. Now, one thing you should be wary of when appraising SRMAs is the presence of publication bias. Now, this type of bias comes about because positive results are more likely to be published than negative results. If only published articles are sought, the effectiveness of an intervention, safety of a therapy, or accuracy of a test may be overestimated, and thus biased. Thoroughly searching for and including unpublished articles is thought to be a measure to avoid publication bias in SRMAs. A caveat to this, however, is that preprints and unpublished articles are not peer-reviewed. Now, the next question under validity is, was the validity of the individual included studies assessed? To be able to answer this, look for an assessment of the methodological quality of the included studies in the review. Many times, SRMA authors assign quality scores based on different scales. Those on therapy very often make use of the Cochrane tool for assessing risk of bias, while those on diagnostic accuracy very often make use of the Quality Assessment of Diagnostic Accuracy Studies tool, or QADAS. So far, no tool specific for SRMAs on harm or prognosis has been devised. Such studies alternatively use the Meta-Analyses of Observational Studies in Epidemiology tool, or MOOS, instead. Now, for purpose of SRMA appraisal, the next few slides will flash important validity criteria questions for assessing the quality of primary studies included in the SRMA. Take note that these are the same questions for validity appraisal that you have already encountered under the topics of therapy, diagnostics, harm, and prognosis. Take note as well that most of the questions under the topic of therapy, which is in the next slide, are similar to that found in the Cochrane tool for assessing risk of bias. Here we have an example of methodological quality assessment using the Cochrane tool for assessing risk of bias. This should appear in the SRMA if the review is on therapeutic interventions. Note the color coding used, where green represents low risk of bias, yellow for unclear risk of bias, and red for high risk of bias. For each of the included studies, the risk of bias in each of the 6 domains in the tool is determined. Then afterwards, a composite bar graph is constructed to present the proportions of studies with low, unclear, and high risks of bias per domain. And here we have an example of methodological quality assessment using the QADAS2 tool. It applies the same concepts as with the Cochrane tool for risk of bias assessment, except that a different set of domains are being assessed here. The next and last question under validity appraisal is, were the assessments of the included studies reproducible? Now, authors of SRMAs have to make decisions on which studies to include, how valid the included studies are, and what data to extract from these studies. Now, these decisions are difficult to make and are subject to human error. To ensure that the assessments are reproducible and unbiased, there should be at least 2 independent reviewers of the included studies. When you appraise an SRMA, make sure to check in the methodology that at least 2 authors evaluated the quality of the included studies. There should also be mention or description of the process to undertake if disagreements occur between the independent reviewers. This usually involves either discussion or conference with a third person. We now move on to appraising the results. We start off with first getting an overview of the SRMA findings. In addition to descriptions of the population and intervention or exposure of interest, all systematic reviews and meta-analyses should present a qualitative summary of the characteristics of the included and excluded studies. These summaries should contain information on the quality of included studies, indicating potential sources of bias. Systematic reviews and meta-analyses should also present a summary of the results of included studies. This is usually presented as a table or forest plot, with results reported separately for each study. In the case of a meta-analysis, There should also be an overall or pooled result. Outcomes can be reported as dichotomous variables, which have only two possible results. For such outcomes, SRMAs can summarize the results as the overall relative risk, risk difference, or odds ratio. Sometimes the hazard ratio, absolute risk reduction, or relative risk reduction will also be reported. For diagnostic SRMAs, on the other hand, the overall sensitivity and specificity are reported. When outcomes are reported as continuous variables with a range of possible results, the difference in outcome is now expressed as a difference in means between the treatment and control. Sometimes, this mean difference is standardized. This standardized mean difference, or SMD, is used as a summary statistic when the studies all assess the same continuous outcome but measure it in a variety of ways. For example, all studies measure depression, but they use different psychometric scales. Here we have a forest plot of the hypothetical SRMA that investigates a dichotomous outcome. Now this figure is lifted from the Painless EBM book. Take note of the text balloons labeling the most important parts of the forest plot. Now each tree in the forest plot represents a study, wherein the square is a point estimate and the horizontal line that runs through it is a 95% confidence interval. The diamond, on the other hand, is a summary or pooled effect of all the studies. The apex is a point estimate, and the ends are the 95% confidence interval. Notice that the width of the diamond is supposed to be the narrowest, since it represents the total sample contributed by each included study. Finally, the midpoint of the x-axis represents the null value, or the value that represents no effect. It is 1.0 for dichotomous variables and 0 for continuous variables. There should likewise be labels on the x-axis that indicate which side is benefit or harm. Here are two forest plots summarizing the results of studies with continuous outcomes. Notice that each treatment arm has a column for the mean and standard deviation of the outcome, besides the total number of individuals. The upper forest plot uses mean difference as measure, while the lower one uses SMD as measure. Now this next forest plot summarizes the results of studies that investigated a dichotomous outcome. Note that the measure used is risk ratio, and for each treatment arm, the number of individuals who attained the outcome and the total number of individuals are indicated. Now, for more details on forest plots, I refer you to the supplemental video I provided under this module. Overall, though, a forest plot can give us information on the following: the number of studies included, the studies with the largest sample size, this is generally represented by the biggest square, the studies with the most outcome events, they generally have the narrowest 95% confidence interval line among the trees in the forest. Which studies show significant benefit? This means that their entire 95% confidence interval line is on the left of the null value. Which studies show significant harm? This means that their entire 95% confidence interval line is on the right side of the null value. Which studies are inconclusive? This means that the 95% confidence interval line straddles the null value and extends far to either side. Which studies show a trend towards benefit? This means that the 95% confidence interval line is to the left and barely touches the null value. Which studies show a trend toward harm? This means the 95% confidence interval line is to the right and barely touches the null value. Which studies show equivalence? This means that the 95% confidence interval line straddles the null value, with both ends of the line near said null value, not extending far away, meaning that this 95% confidence interval line is narrow. And lastly, whether there was heterogeneity in the results. We will touch on heterogeneity in a bit, but a rough way of assessing it is by dropping a vertical line through the lines and see which ones do not intersect or overlap with said vertical line. The next question under results appraisal is, how precise were the results? Again, we refer to the 95% confidence interval of each study included in the SRMA, and the width of the diamond or pooled estimate. As expected, the overall interval estimate of the diamond is usually narrower than the interval estimate around the individual studies. This gain in precision is the main reason why we do SRMAs in the first place. As with individual trees of the forest plot, look at the direction or position of the diamond's 95% confidence interval as well. Is this pooled estimate showing us significant benefit or harm, or simply a trend towards? Is the overall pooled result inconclusive, or is there equivalence between the treatment arms? Now, this next question involves the concept of heterogeneity that I mentioned earlier. Between included studies, there may be subtle variations in the population, exposure, intervention, and outcome. This is what we call the between-study variability, which is essentially the heterogeneity. Even if the P, I or E, and O are similar, the study methodologies used may be slightly different. Generally, when the heterogeneity is too great, reviewers become hesitant to perform a meta-analysis and estimate the average results. To determine whether the results are similar enough to justify combining them, check whether the authors assess for heterogeneity. Generally, heterogeneity is assessed using two statistical means. The first one is a chi-square test. This yields a p-value which, if less than 0.10, indicates significant heterogeneity. This means that differences between studies cannot be attributed to chance alone, and that there are more likely methodological differences involved which have to be investigated and described further by the reviewer supposedly. The second statistical method is the I-squared statistic. While the chi-squared test indicates presence or absence of heterogeneity, the I-squared statistic provides information on how big that heterogeneity is. I discuss this in more detail in the supplemental video, but a value greater than 40% generally represents substantial heterogeneity. When heterogeneity is substantial, it is generally recommended that what is called a subgroup analysis is performed. Because differences in P, I or E, O, or M are the most common sources of heterogeneity, the reviewers can analyze subgroups of these PCOM-PECOM components within which the results might be homogeneous. Sex and age groups are very common subgroups that are investigated. This is discussed in more detail in a supplemental video. A caveat to this is that too many subgroup analyses raise the possibility of accidentally finding benefit in one subgroup, particularly if such analyses are unplanned or not considered beforehand in the methodology. We call this data dredging. If we overanalyze after the fact, then we are bound to find something significant. Keep in mind that, more often than not, subgroup analyses tend to be underpowered in terms of sample size. This is why we don't simply take subgroup analysis results as gospel truth right away, unless the magnitude of effect size is convincingly large enough. Rather, we should regard them as points for future investigation via further research. For that matter, subgroup analyses are most credible when the following conditions are satisfied. They are planned beforehand, there are not too many of them, and the results are biologically credible. If subgroup analysis fails to identify a credible source of heterogeneity, there is no reason to insist on presenting a quantitative estimate of overall or pooled effect. Instead, the authors should resort to a qualitative discussion and summary of results. After appraising results, we assess the article's applicability with our patient. Assessing the applicability of the results of SRMAs follows the same principles described with therapy, diagnostics, prognosis, harm, and CPGs. And by that, we apply the mnemonic scraps, which give us an idea of which factors may affect applicability of the estimates presented by the article being appraised. If the overall results of the SRMA are not directly applicable to your patient, it may not necessarily mean that it is entirely useless. There may be actually valid subgroups that were analyzed that fit your patient's characteristics. But as discussed earlier, the results derived from a subgroup analysis should be interpreted with caution and should prompt you to search for articles specific for the subgroup your patient is in. Now we come to the last part of the appraisal process, individualizing the results to your patient. Application of the results of the SRMA to an individual patient or patient group depends on what type of research question that SRMA addresses. If the question is on therapy, find out what the patient's baseline risk is based on his or her clinical characteristics. Afterwards, look for the pooled relative risk reduction, or hazard ratio from the article. which you can then use to derive individualized measures, which are the absolute risk reduction and number needed to treat. With diagnostics, on the other hand, you need to first estimate your patient's pre-test probability of disease. You then obtain the pooled likelihood ratio for a test result from the SRMA, which will then use to determine that particular patient's post-test probability of disease. You've learned previously that with research questions pertaining to harm, the appraisal is somewhat similar to that of therapy with some differences. You also gauge your patient's baseline risk. You then look for the pooled odds ratio, risk ratio or hazard ratio from the SRMA, which you then use to estimate the absolute risk increase of that patient, as well as the number needed to harm. Finally, with prognosis, take note of your patient's characteristics thought to be related to the outcome of interest. Then from the SRMA, look for the pooled event rate given patient characteristics or a predictive score, and from there determine patient-specific probabilities of developing these outcomes. And that is it for this video lecture. Here are my references. I highly recommend that you watch the supplemental video I provided as well. Thank you very much and see you in the upcoming workshop.